Hello, my name is Fenet Demissie. I'm a third year student at Lehigh University and I work in the Chow Lab. My project is titled Spatiotemporal Presentation of Bioactive Peptides on 3D printed scaffolds, and the issue that we're trying to address is osteoarthritis. So osteoarthritis is a debilitating disease that is caused by the degeneration of the osteochondral unit, which includes the articulating joints. And the current gold standard treatment for osteoarthritis is total joint replacement. However, a lot of younger patients tend to outlive their transplants and require revision surgeries. As an alternative, exogenous growth factors are commonly used to enhance osteochondral tissue repair and regeneration, but whole growth factors or naturally occurring growth factors tend to have shorter half-lives and they are required to be used above physiological levels for regeneration, which can make them costly and dangerous to the patient. So our lab is looking at an alternative way, growth factor mimetic peptides, which mimic the chemical and mechanical structures of naturally occurring growth factor. In this project, we are using transforming growth factor beta 1, which is key in chondrogenesis or cartilage formation. From literature, we found that the LIA and AK sequence can mimic the beta bend in the naturally occurring TGF beta 1 growth factor, and it can mimic the activity as well. We modified this LIA and AK sequence with DBCO or dibenzoyl and this is a click chemistry group that can react selectively with azide, and we also modified this peptide with rhodamine, which is a fluorophore and can be used to visualize the peptide location. So that peptide with the DBCO and the fluorophore modifications was the first part of this study. So the second component of this study is the PCL azide scaffold. So in order to be able to 3D print this scaffold, we first have to make our PCL azide conjugate. So how we make it is we first start with a low molecular weight 14 kDa polycaprolactone. So polycaprolactone is a biodegradable polymer that we use to 3D print, and we take this 14 kDa low molecular weight PCl and we modify it with PEG azide to be able to make our PCL azide conjugate. We then take this conjugate and we react it with an 80 kDa high molecular weight unmodified PCl. So 20 mg per mil PCl azide conjugate is dissolved with 370 mg per mil unmodified high molecular weight PCl in a really highly volatile solvent called HFIP. And this mixture is then shaken, made into an ink, and it is 3D printed to produce PCl azide scaffolds. So the way we attach our peptides to our PCl azide scaffolds is by using bioorthogonal click chemistry. Bioorthogonal click chemistry refers to these really fast and highly selective reactions that can take place in the presence of cells without interfering with the cellular activity. So we have DBCO on our peptides and we also have azide on our scaffolds. And DBCO and azide are two click chemistry groups that can react with each other really selectively and efficiently. So this way we can present our peptides on the surfaces of our scaffolds. So in order to do a proof for this hypothesis, we did a proof of concept study with PCL only scaffolds and PCL azide scaffolds. And we took these scaffolds and we labeled them with DBCO containing TGF beta 1 peptide. So when we labeled both PCL only scaffolds and PCL azide scaffolds, only the scaffolds that contained the azide click chemistry group fluoresced, which confirmed our hypothesis that the azide and the DBCO do react selectively, and our peptide would not just attach to PCL by itself. So we used a fluorescence microscopy method to be able to quantify and visualize fluorescence. So to conclude, in this study, we synthesized the TGF beta 1 mimetic peptide, and we modified it with rhodamine and also DBCO. And we also successfully synthesized our PCL azide scaffolds, and we 3D printed with them and also fluorescently labeled them. Future work is going to be to repeat this study and demonstrate the peptide attachment in the presence of cells over time. So the over time component will be the temporal part of our spatiotemporal study. So we are also synthesizing a BMP2 mimetic peptide, and this peptide is going to promote bone regeneration, and we will be using two distinct bioorthogonal chemistries. So we have our DBCO azide chemistry in the current study, and the BMP2 study is going to use a completely separate click chemistry group. And our goal is to be able to spatially and temporally deliver both the TGF beta 1 and the BMP2 mimetic peptides during cell culture for a stable osteochondral regeneration. Thank you.